Good morning, students. I'm Dr. Samrina. Today we are going to discuss the protein trafficking to the endoplasmic reticulum, that is, rough endoplasmic reticulum. As we know, the eukaryotic cell has 10% of its volume occupied by these endoplasm reticulum. Endoplasm Endoplasmic reticulum occurs in three main structures. First is the cisternae. These are long flat sac-like structures having diameter of 40 to 50 micrometers, and the another form of endoplasmic reticulum are in the form of vesicles. These are unbranched membrane bound tubules with the diameter of 25 to 500 micrometers. Then the third form of the rough endoplasmic reticulum are in tubules. They are with branched structure having diameter of 50 to 190 micrometer. As we know, this endoplasmic reticulum is the largest organelle, or this is the largest organelle present in the eukaryotic cell, which are having continuous membrane with the nucleus on its one side, and on the other side these endoplasmic reticulum are attached with the at many points with the Golgi bodies. The whole membrane system is called as endomembrane system. We are having two different types of endoplasmic reticulum. One is the rough endoplasmic reticulum, and the second one is the smooth endoplasmic reticulum. So, on the rough endoplasmic reticulum or the rough endoplasmic reticulum are involved in the protein synthesis, and the smooth endoplasmic reticulum are involved in the lipid metabolism. The first demonstration of role of endoplasmic reticulum in protein processing was carried out by George Plade and his co-workers in 1960. So in the above diagram, we are going to study about the Plade's experiment. They were able to study the pathway taken by the secretory proteins which were translated on the ribosomes of endoplasmic reticulum by simply labeling newly synthesized proteins with the radioactive amino acids. The location of the radio labeled proteins within in the cell was then determined autoradiography by the autoradiography by following the pathway of these radioactive labels in the cell. By observing the pathway of these proteins, we observe the pathway via the with the help of radioactivity. Then we were able to identify how proteins are transferred from rough endoplasmic reticulum to the Golgi bodies, then from Golgi bodies to the further to the then from Golgi bodies further either to the lysosomes or to the outer cell, or these proteins are then sent to the outer surface, or they are having third path, they make the part of the plasma membrane. As you can see here in the diagram, that we are having radioactive label proteins. Then in the second, these from the within the 7 minute of chase, these proteins were able to move from rough endoplasmic reticulum to the Golgi bodies. This is shown with the part. Then from Golgi bodies, these proteins were transferred further, either outside the cell, or they were forming the early endosomes or the lysosomes present in the cell. The whole pathway, they call this as secretory pathway. The whole pathway, they describe this as a secretory pathway in which they said that the proteins are translated on the rough endoplasmic reticulum. Then these proteins are inserted into the lumen of the endoplasmic reticulum where they get post post translationally modified. Then from rough ER to the Golgi bodies where they get further modified. Then from Golgi bodies these proteins are secreted outside the cell. Later on, it was further investigated not only secretory proteins follow this path, but the plasma membrane as well as the lysosomal protein also follow the same path. And then to their final destinations, means they follow from rough endoplasmic reticulum through the Golgi bodies, then to the final destination. Some proteins travel initial steps. There are some proteins which we have seen that they follow the initial step, that is the proteins are translated on the endoplasm on the rough endoplasmic reticulum membrane. Then these proteins are inserted into the lumen of the endoplasmic reticulum. Then there are some proteins which are the permanent residents of the lumen of the endoplasmic reticulum, or they will further move to the Golgi bodies where they are the permanent residents of the Golgi bodies. To follow the secretory pathway, proteins have to enter into the Golgi bodies. So at the endoplasmic reticulum, we can say that this is the major branch point for trafficking of the proteins. Whether protein has to send further to the Golgi bodies or these proteins will be the part of the endoplasmic reticulum. So here in this slide we are having the fate of the mRNA. There are mRNA which are getting translated in the cytosol. From the cytosol, these proteins are then transferred to the nucleus or to the mitochondria or to the chloroplast or peroxisomes. But the proteins which are translated on the membrane of the endoplasmic reticulum, these proteins are sent to the either they become the part of the plasma membrane or they become the part of or they become the secretory vesicles or they become the lysosomes of the cell. So if the proteins are translated in the cytoplasm, they are having different destiny than the proteins which are translated on the bound ribosomes which are present on the rough endoplasmic are having different destiny. When proteins are approaching towards the rough endoplasmic reticulum, in the cytosol only, the ribosomes get attached to this mRNA, which is going to be translated on the surface of the endoplasmic reticulum. So the whole structure, ribosomes along with the mRNA, is called as polysomes. That is, proteins or the mRNA along with the ribosomes are then translated on the ribosomes which are present on the surface of the endoplasmic reticulum. Then they are sent into the lumen of the endoplasmic reticulum for post translation modifications. So to move mRNA towards the endoplasmic reticulum, they should have signal sequence, and they scientists have observed that they are having signal sequence of about 20 amino acids, which includes the stretch of hydrophobic residues usually present at amino terminal of the polypeptide chain. Means at its amino terminal we are having 20 amino acid residues which are hydrophobic in nature. As the polypeptide chain emerges from the ribosome, the signal sequence, as we can see in this diagram, the signal sequence is recognized by a certain particle. This particle is called as signal recognition particle. This particle is called as signal recognition particle. As you can see, when mRNA is approaching to the surface of the endoplasmic reticulum, so ribosomes get attached on this mRNA. The whole structure is called as polysomes. When they reach at the near to the surface of the endoplasmic reticulum, there is a one particle which is called as signal recognition particle. This signal recognition particle will bind to the both ribosomes as well as to the signal recognition sequence of the growing polypeptide chain and it shows it way towards the membrane of the endoplasmic reticulum where this sig- where this signal recognition particle binds with the signal recognition particle receptor binds with the signal recognition particle receptor which is present on the outer which is present on the membrane of the endoplasmic reticulum. The binding of receptor, the binding of this particle to its receptor when this when this binding of receptor releases SRP from both ribosome as from the signal sequence. That is, when the SRP gets binded or it get attached to its receptor, then this SRP protein get released from both from the ribosome as well as from the signal sequence. The ribosome then binds to the protein translocation complex. As you can see here, translocators are there. So ribosomes then bind to the protein translocators and and insert signal sequence into the membrane channel membrane channel into the membrane channel is the same diagram but with different look, but we are having the same, but this is the same diagram which I have showed earlier. The channel is thought to be made up of transmembrane proteins. The channel is thought to be made up of transmembrane proteins, and they have called these proteins as SEC61 proteins. SEC61 proteins. In yeast and mammals, SEC61 proteins are closely related to the plasma membrane proteins that translocate secretory proteins in the bacteria, demonstrating a striking conservation of protein secretion pathway in prokaryote as well as in eukaryotes. Means similar type of proteins have been observed in yeast mammals, and this type of proteins are also present in the bacteria. The ribosomes on SEC16 translocation complex resumes and allow the insertion of growing polypeptide chain into the lumen of the endoplasmic reticulum. So first there is the insertion of signal sequence into this channel. So as translocation proceeds, the signal sequence is cleaved. The signal sequence is cleaved here by the signal peptidase, which is present on this channel at its inner side. So there is the presence of signal sequence which cleaves this signal sequence from the polypeptide chain. The enzyme which is responsible for the cleave of signal sequence from the polypeptide chain is called as signal peptidase. So some proteins are translocated onto the free cytosolic ribosomes and come into the lumen of ER for post-translation modifications. We are having different set of proteins which are translated on the free ribosomes which are present in the cytosol. So they need to get modified. So they will come onto the lumen of endoplasmic reticulum to get modified. That is, they have to be modified by post translational modification. So they will again come onto the surface of the endoplasmic reticulum. These protein does not require SRP particle. They don't need signal sequence particle. Instead, their signal sequence are recognized by the receptor different receptor proteins. These receptor proteins are called as Sec62 and Sec63 complex. The protein which are meant for endoplasmic reticulum are are getting recognized by Sec61 protein complex. So this is a direct transportation of mRNA from nucleus onto the surface of the rough endoplasmic reticulum. But the proteins which are getting translated in the cytosol, these proteins need now post translational modification. For their post translational modification, they will come onto the surface of the endoplasmic reticulum. So there we are having different complex which will recognize this polypeptide chain. This complex protein complex is called as Sec62 and Sec63 complex, which is associated with Sec61 complex of endoplasmic reticulum. Here you can see that we are having two channels together, Sec61 complex and Sec62 and Sec63 complex. Chaperones present in the cytosol, as we know, chaperones, these are also, we can call them as heat shock proteins as well. These chaperones present in the cytosol help to maintain the growing polypeptide chain in the primary structure. Means these chaperone help to maintain the primary structure of the growing polypeptide chain. Then only these polypeptide chains can enter into the Sec61 channel. Another chaperone which is present inside the endoplasmic reticulum is called as BIP. This BIP pulls the this BIP pulls the growing polypeptide chain through the Sec61 into the lumen of the endoplasmic reticulum. So this BIP acts as a driving force for post-translational translocation of proteins into the endoplasmic reticulum. But I had said BIP is an important for polypeptide as it drives the post-translational translocation of proteins into the endoplasmic reticulum. So now we are having what we are having now protein in the lumen of the endoplasmic reticulum. So now these proteins need to get fold first, then after folding they need to get processing. So the next is the protein folding and processing inside the lumen of the endoplasmic reticulum. To attain the correct three-dimensional conformation, these proteins need to assemble need to get assembled, or different polypeptide subunits are coming together which form the three-dimensional conformation. So there is some modifications, mm, covalent modifications occur across the endoplasmic reticulum membrane, or we can say directly the assembly of polypeptide in the multi-subunit, and covalent modifications occur across the endoplasmic reticulum membrane or within in the lumen of the endoplasmic reticulum membrane. So first process is the removal of signal sequence by the signal peptidase. This was the first function. Then the main function of endoplasmic lumen is to catalyze the folding. Now what is happening when the proteins are in the lumen of the endoplasmic reticulum? They have to they have to catalyze the folding and assembly of newly translocated polypeptide, and this folding is carried out, or this proper folding of this polypeptide chain is taking help of the chaperone HSP70. This folding is carried out with the help of HSP70. This HSP70 is a family of chaperone called BIP. BIP allow protein assembly correctly. This what is a, now we are having BIP there. We are having many important functions of the BIP. First, it allows the growing polypeptide chain into the lumen of the endoplasmic reticulum. This BIP allows, the second important point of BIP is BIP allows protein to assemble correctly. Then when the protein assembly is correctly, then BIP get released from these proteins so that further further then transported to the Golgi bodies. In case where in case if there is misfolding of the proteins in the lumen of the endoplasmic reticulum, that time BIP remains attached to these polypeptide chains and this protein is retained within in the endoplasmic reticulum, or rather we can say they get degraded by lysosomes. So what happens, these proteins via the bulk transport system are sent from endoplasmic reticulum to the Golgi bodies. Then from Golgi bodies they are sent to the lysosomes where these proteins along with the BIP get degraded. So this was about the protein folding. This was about the protein folding. One important process also carried out is carrying in the lumen of the endoplasmic reticulum is that when protein get assembled, there is the formation of disulfide bond between the cysteine residues of the polypeptide chain, which plays an important role in the proper assembly to make the primary structure into the three-dimensional structure. The lumen of the endoplasmic reticulum is the lumen only where these disulfide bond formation takes place, where these di- disulfide bond formation takes place. So, in the lumen of the endoplasmic reticulum, this the bond formation is catalyzed by one enzyme. That enzyme is called as disulfide isomerase. That enzyme is called as disulfide isomerase. So, this was about the transportation of proteins into the endoplasmic reticulum.